I'm going to talk about the structural analysis of the compendium of RNA-binding proteins, and in fact, touching on many questions that Chris Burge raised in his keynote yesterday, only using a different data sets, RNA-compete. So as we may all know, protein RNA binding is very important for many roles, and has many roles and important for many processes in the cell, including the capping of the mRNA, the splicing of the mRNAs from its raw form into its mature messenger RNA form, the 3' end cleavage, the transportation from the nucleus to the cytoplasm. It has an effect on the stability, such as increasing the stability or increasing the degradation rate, and also some effects on the translation. So for this aim, we want to measure and to have good models to predict protein RNA binding. And most of these interactions and the binding to RNA are mediated through both the RNA sequence and the structure. So the proteins would recognize specific RNA sequences in specific structural context. And in the next slide, I'm going to describe and tell you what do I mean by structural context. So in many studies, as well as this one, when I'm talking about RNA structure, I'm talking about the secondary structure. And this is mainly, since this can be predicted in feasible times computationally. An RNA secondary structure is defined as the base pairing of nucleotides, mainly the canonical base pairs G, C, and UA, but you can also have base pairing of some non-canonical base pairs. And in many cases, it's not the specific base pairing that matters for the protein binding, but it is the structural context that is induced by that base pairing. So here I'm showing an example of 6 different contexts, and it may be that the protein would like to bind a specific sequence in a hairpin loop, or a paired region, a bulge loop, internal loop, external, or multi-loop. So these are the structural context. Now, to make things more complicated, a protein does not fall necessarily into just one structure, but it may fall to an exponential number of structure. And we need to take this into account. So one way to represent the ensemble of possible structure is using aggregated probabilities. And by that, I mean that for each nucleotide, we now consider a probability of being in any one of these structural contexts. So for example, we can measure them using an experiment, such as IC shape, and get a probability of a nucleotide to be paired or unpaired. Or we can run RNA-PL fold from the Vienna package and get the distribution over 5 different structural contexts. So this is how we represent the ensemble of structure that an RNA molecule may be found at. To measure protein RNA binding, this can be done using the CLIP protocol to measure the binding in vivo. And in this protocol, the protein is cross-linked with the RNA using UV lightning, and then there are several steps that the RNA and the protein go through until at the end we take out the bound RNA sequences of specific length. Then we can sequence those sequences, the bound ones, and then perform some peak calling to get the peaks, the binding sites that were bound on the transcriptome. Very similar to the ChIP-seq protocol for protein DNA binding. But there are several disadvantages in this protocol. So first, if we are interested in learning the structural preference of the protein, we need to have the structure of the mRNA. And the prediction accuracy of structure on mRNAs in vivo is not that accurate. It's very difficult to predict the structure of long mRNAs, which may span thousands of nucleotides. Moreover, we may find cofactors, meaning proteins that bind together as a complex, or competing proteins, which would compete for the same binding site. There might be technological biases. Some were reported in previous studies. And we need to measure the RNA expression to see how much RNA was available for the protein to bind. And so to get a cleaner view of the intrinsic binding preferences of protein, several protocols were developed to measure protein RNA binding in vitro. One of them is RNA-binding from Chris Burge lab that was described yesterday. And the other one is RNA-compete from Tim Hughes lab from the University of Toronto, which I'll use in this study. So in RNA-compete, 240,000 RNA probe sequences are available for binding. And the mathematical property of all of these RNA sequences together is that each RNA 9-mer appears at least 16 times. So very similar to protein binding macroarrays where every 10-mer appears once. Here we also have some guarantee of the coverage of 9-mers. Another property of the RNA probes is that they were designed to be unstructured, under the belief that most proteins would bind accessible regions, meaning unpaired regions of the RNA. So the protein may bind any of the sequences, and the bound ones are pulled into a microarray, and by hybridization to complementary sequences, we get a proxy for the binding intensity of the protein to those sequences. And the final output is the set of sequences and the binding intensity. So the advantages is that now we are using much shorter RNA sequences. They are between 30 to 40 nucleotides, and the prediction, the computational prediction of the structure is much more accurate. Moreover, there is only one single protein in the assay, so there is no competition and no binding as a complex. Hopefully, there are less technological artifacts, as this is an in vitro assay, and it's easy to measure the background signal. So one of the computational challenges that rise from this data is to infer the binding preferences of the protein. And previously, RNA context was developed to represent the preferences using a position weight matrix model, which you see on the left side, as well as to keep 5 parameters for the structure preferences. We have a work that was recently presented at ISCB and published in Bioinformatics, where we extended this model into a k-mer-based model, where we keep a sequence score for each k-mer, as well as different structure preferences for each of these k-mers. So this allows for the modeling of much more complex binding modes. So it allows for a modeling of dependency between position in the binding sites, as well as different structural preferences for different k-mers. And we have shown that we outperform the state-of-the-art, both in sequence models, deep bind, and in structure-based models, RNA context, when we compare on the RNA-compete dataset. So now what is the challenge that we want to tackle in this study? So 3 years ago came out a Nature paper from Tim Yu's lab, a Ray et al. Paper that measured the binding of 205 proteins. So this was the largest scale data sets of protein RNA binding measurements. But in this study and in following studies that look at this data, they only focused on the sequence preference of these proteins. Now that we have the new algorithm that we termed RCK, we can infer the structure preferences from these data. Now it's the first time that we can ask what are the common structural preferences of the 205 proteins in this data set, and maybe conclude something even broader about RNA binding proteins. So this is what we went and did. So first I wanted to convince you and myself that indeed we do see some structural variability in the data. And if you recall, I told you that in the RNA-compete assay, they used unstructured RNAs. So we wanted to measure how unstructured are these RNAs. So we compared the 240,000 RNA-compete probes to random probes, and we used the same definition of probability of being structured. And as we see, random probes reach all the way to the probability of 1, but we also see that the RNA-compete probes have some probability of being structured, and in fact, more than 75% of them have a probability greater than 0 of being structured. So meaning there is some structure in the data. Another way to look at it is to predict the probability of nucleotides to be paired. And in this case, we again see that the RNA-compete probes span the whole range between 0 to 1 in probability of being paired. So many of the nucleotides are paired in those unstructured RNA probes. Moreover, we wanted to see if we have structural variability for different 5-mers. And why do we analyze 5-mers? Because these can be typical lengths of a protein RNA binding site. So we measure the standard deviation of the average 5-mer pair probability. We see that the variability is not as high as in random probes, but still we see high probability. And when we want to test how well this variability correlate with the binding affinity, we took the example of the HUR protein, which is known to bind the U stretch and prefers to bind it in unpaired context. When we look at the correlation between the binding intensity of the different probes and the average paired probability of the U stretch in them, we get a significant anti-correlation. It's not a very high correlation, but maybe we can utilize this information, as I said, we did utilize it in our algorithm, to learn the structural preferences of RNA-binding proteins. So next, we went and asked, what do RNA-binding proteins like to bind overall? For this aim, we took the top 5 mer, what we term the consensus sequence, the consensus binding site of each RNA-binding protein, and we measured the log ratio between the binding in context 1 to another context 2. So first, we looked at the context of unpaired compared to paired. And what we see is that overall, almost all proteins prefer to bind in unpaired regions. And this is in agreement with the main assumption that RNA-binding proteins prefer to bind unpaired regions. And we have a few exceptions. These may be either error in the models or few exceptions in the protein world for proteins that prefer or can tolerate binding to paired regions. Moreover, we went and looked if there is a specific preference in binding to unpaired regions between loop and external regions, where loop are regions which are encompassed in a stem, as opposed to external regions which are just out there in the RNA sequence. And we see that there is some preference to loop regions, although many proteins are quite indifferent and may bind in either of these contexts, in a loop or an external region. We don't see any division to different domains. And we believe that this is due to the fact that these domains were defined in a very broad manner. There is little sequence similarity for the proteins in these domains. Next, we wanted to test how much do we gain by using the structural information. So if you recall, I told you about the model that represents the binding preference. It keeps sequence scores as well as structure scores. So now we compare the prediction of binding just using the sequence scores as opposed to using sequence and structure scores. So first we test how much we gain on in vitro data, RNA-compete data, where we train for each data set, for each protein, we train on one half and we predict on the other half. And we see an improvement across the board. So all proteins gain from having this additional structural information. And the advantage that we see, the increase in average Pearson correlation, is striking. It goes from 0.31 to 0.46. Now we went and we asked, how well do these structural preferences that we learn from in vitro data correlate with in vivo data? So we took the eClip data that Chris also mentioned in his keynote yesterday. This is an improved protocol for clip experiments. And we tried to predict the binding to the peaks that were found in these in the eClip experiments, as opposed to negative sequences which we took downstream. And we see, again, a significant improvement in the binding prediction, but the increase in AUC is much milder. It is not profound, although it is significant. We believe that the effect is not as strong due to the noise that we find in vivo experiments, and the fact that the structure prediction is not as accurate. And it might be that the models that were trained on the in vitro data have some overfitting to the technological artifacts of those experiments. Here I'm showing one example for one of the proteins which benefited most from using the structural information, going from AUC of 0.75 to 0.81. And this is the first discovery of its structural preference to multi-loops and external regions. So last, we concluded with performing a transcriptome-wide prediction of protein-RNA interactions. We used the algorithm and took 102 human proteins that were available in the compendium of RNA-binding proteins and predicted the binding on the human and mouse transcriptome. We include in the output the Z scores and conservation scores, and this is available through this web address, so you're welcome to download the data, and I would welcome any suggestions and feedback on how to improve that. Future directions would mainly try to bridge the gap between in vitro and in vivo. So one way to do it is to use experimentally measured RNA structure as opposed to predicted one. To explore the tissue-specific RNA binding, the structure may change between different tissues, and to improve predictions by transcriptomic context. So to summarize my talk, I've shown that structural variability does exist in unstructured data, in unstructured RNA, and it is correlated with protein RNA binding. Most of these proteins bind unpaired regions, and many of those prefer to bind loops. I've shown that we get a significant improvement, both in predicting in vitro binding and in vivo binding for this data. And we performed a transcriptome-wide prediction of the RBPOM, which is now available for the community. So I thank Professor Berger and the lab members and you for the attention.
Really nice talk, obviously. We have similar interests. I was curious, so you mentioned that most of the proteins like to bind hairpin loops, for example, or other unpaired regions. Did you find any proteins that prefer bulged RNAs or base paired sequences? And if so, were there any surprises there or interesting?
Well, yeah, there were a few exceptions in the plot that I've shown here, right? Binding to paired regions, and I don't have another validation set. Maybe if I compare to RNA bind and seq for the same proteins, I could validate that.
Yeah. Welcome to it's online.
Yeah, so that would be a good validation to see if really there are a few exceptions. I know that there is the example of Staufen, which binds paired regions, but we don't have that in this data set.
I think probably both our group and Tim's group probably avoided those proteins.
Okay, yeah.
Yeah,
and I think I saw a few examples of protein which prefer bulge loop, but I haven't gone into the details of these proteins. So
I think that'd be interesting to confirm examples.
Yeah, so the models are available online. Yeah, so I've put them on the website. Yes.
I have a quick question. In the in vivo comparison between the sequence and sequence plus structure, it looked like most of the points were on the diagonal. The off-diagonal ones were on the left. And so it looked like those in which the sequence alone does very badly, that's where the sequence plus structure gives a big improvement. And that's what gave you the good p-values. Do you have any comments on what was going on there?
Well, there are a few more examples. For example, H and RNPK improved from 0.75 to 0.81, so that's like above, on top of the diagonal. Okay. But I agree that, so I don't know what's going on in those experiments. It is, I mean, surprising that you go from AUC that is maybe below 15 to something that is higher than random due to the structure preference. But I don't have an answer on what exactly is going on. And I believe there is a lot more work that we need to do in the community to bridge this gap, just like in protein DNA binding. Yeah. So now we have to do here. Yeah.
There are no other questions. Let's thank Yaron once again.